Hello everyone, and welcome to the first pre-lecture for the first flipped classroom activity, where we're going to deal with cellular metabolism. So we will begin here with some of our reactions that we talked about in our pathways in terms of transport reactions and other things, biosynthesis and fueling reactions to come later potentially. So first let's talk about transport reactions, where we think about bringing substrates into the cell, growth substrates, substrates that the cells need to survive. How do they get in? So we can have facilitated transport, which involves proteins that are present in the cell membrane and can help shuttle things across. And when I talk about facilitated transport, we're talking about going down the concentration gradient into the cytoplasm or the cytosol of the cell. So essentially we have a carrier that will bind from what is called the abiotic phase, or basically outside the cell, to this carrier protein. We have this substrate binding to the carrier protein to form a carrier substrate complex. And at first, that carrier substrate complex is on the outside of the cell, but then it traverses through the membrane to the inside of the cell, where the carrier substrate complex can then dissociate to basically spit the substrate out into the cytoplasm with the cytosol, and then the carrier becomes free to bind and carry more substrate into the cell. So when we're talking passive transport here, we're talking the principle of sending these things down the concentration gradient. So when we do passive transport in cells, we have the option to have no protein facilitator because we are going down the concentration gradient. Substrates will potentially just diffuse through the membrane to go into the cytoplasm. Although typically the solute must be either hydrophobic enough or small enough to be able to traverse the cell's membrane bilayers. So they either have to be like fatty acids to be hydrophobic enough, or they have to be small molecules like oxygen, like carbon dioxide, and things like that, to just be able to traverse the membrane without any kind of help from a protein that would potentially do facilitated diffusion. So these facilitated diffusions are mediated by either channels or proteins that say open and close in order to help bring solutes down the concentration gradient across the membrane bilayer and into the cytoplasm. So these we start at an area of high concentration outside the cell in all cases, and will then diffuse into the cell cytoplasm, which is an area of low concentration. So if we're talking about going through the membrane, we're talking about the membrane bilayer being the solvent. This is not necessarily strictly the case when we're talking about facilitated diffusion, because we are traversing the substrate through what little solvent there might be in a carrier or a channel protein. So gradients are responsible for a whole lot of things in the cells. Responsible for bringing nutrients in, getting rid of waste products, things like that. But they're also responsible for those fueling reactions to make ATP. So ATP is produced by transporting protons or hydrogen ions down the gradient into cells. So essentially a proton will go into the electron transport chain and get transported out of the cell. As the electron then gets moved down the chain, there will be a gradient built up, a proton gradient, and then down the gradient will take the proton from outside to inside of the cell, which is mediated by this F1, F0 ATP synthase. Again, it goes down the concentration gradient and it goes into the cytoplasm, thus producing ATP from ADP and phosphate, thus giving the cell more energy that it would need to do its biosynthetic reactions. So cells produce the gradient by transporting protons out and then transporting them in, they can take advantage of that gradient and make ATP. They can make energy. Okay, so let's talk a little bit about pH, because when we're talking about protons, we kind of have to talk about pH and acids, amino acids specifically. So when we dissolve amino acids in water, they don't necessarily exist in their neutral form. There are two ends to each amino acid, a carboxylic end and a amino end, and the pKa of the carboxylic acid group is around 2, while the pKb, or the basicity constant of the amine, is around 9.5. And if we're at a pH of 7, which certainly you would be if you were a cell cytoplasm, the amino acid will exist as a zwitterion, so it'll have both positive and negative charges on the same molecule. It will look kind of like this, where we have a positive charge on the amino end because the amino end is protonated, and a negative charge on the carboxy end because the carboxy end is deprotonated. And that makes a zwitterion with a positive and a negative charge, and those charges do cancel each other out. So if we can look at amino acids at different pHs, they look different at pH 1 versus pH 7 versus pH 14. At pH 1, so we're talking about that pKa of 2, we are protonated on the carboxy terminus, the carboxylic acid end, but we are also protonated on the basic side, on the amino terminus. At pH 7, of course, we have that zwitterion, and at pH 14 we have a negative charge because we are deprotonated on the carboxy end as well as the amino end. And so pH 14 highly basic, and we are deprotonated here, whereas in pH 1 we're highly acidic, so we are protonated in both ends. So what we're going to consider for the flipped classroom activity is phenylacetate, or phenylacetic acid. So this is a product of phenylalanine degradation. Phenylalanine is amino acid. It's also the degradation component of lignin. So you may have heard a lot in recent years about using things like lignocellulosic biomass, which is essentially dead plant matter that either comes from, say, corn cobs or leaves and grass and things like that, that can be degraded, hopefully to carbon sources to feed bacteria to make certain bioproducts, things like biofuels, bioplastics, and things of that nature. It's also a degradation component of styrene and ethylbenzene, phenylacetate is. Both styrene and ethylbenzene are environmental contaminants. So, again, we have either dissociated acids, or phenylacetate, and undissociated acids, or phenylacetic acid, and we are trying to get them across the cell membrane. Now, typically there's probably not a dedicated transport protein with which to get these substances across the cell membrane. So perhaps we have to generally rely on diffusion through the cell membrane. And what's the best way to do that if we don't have a transport protein? Well, both dissociated and undissociated acids can cross the plasma membrane by diffusion, but undissociated acids have a much larger solubility because they don't have that negative charge like the dissociated acids do. They have a much larger solubility in the membrane, and so can thus traverse the membrane more rapidly. So we can get more phenylacetic acid into the membrane and thus into the cytoplasm compared to the phenylacetate coming into the membrane as well as the cytoplasm. We can get, per unit time, we can get more of the undissociated acid through because it lacks that charge. And once in the membrane, the organic acid has crossed the membrane, so the dissociation will likely occur then. So it's not going to remain undissociated. It'll thus dissociate to phenylacetate and proton. Carboxylic acids have a negative charge at pH 7, and the cytoplasm is going to be around pH 7. So thus you will get that dissociation. and that'll leave a free proton floating around, which could potentially acidify the cytoplasm if something is not done. So there are two things that concern us: the acid getting into the cytoplasm, but they have a harder time getting out, which is good if we want to use it as a carbon source. But the cytoplasm itself has protons floating around, and we must avoid acidification of the cytoplasm, as I just mentioned. So we can reverse the ATP gradient to pump some of those protons out, but we've got to spend ATP. That's the one downfall here. So if we have a plentiful amount of protons in the cytoplasm could become acidic. The cell must pump it out to avoid this acidification. So the F1, F0 ATPase, which I talked about earlier, the ATP synthase, it does this work. It's going to act in reverse, so instead of making the ATP like it usually does in cases of pumping protons back into the cytoplasm, getting the ATP, it's breaking the ATP. It's using up that ATP to make ADP and phosphate just to get protons out of the cytoplasm and into the extracellular space. Okay, so you have an assignment here regarding phenylacetic acid. We're going to just design an organism, theoretically, that has phenylacetic acid degradation pathway inside of it. And the cell should be able to eat phenylacetic acid. And doing so should provide the cell with enough ATP to at least come out on top. It may not be the best carbon source, but at least it would be a carbon source. So we have a ton of phenylacetic acid, say we have a ton of those contaminants, a ton of lignin, something like that. We want to get rid of some phenylacetic acid. So consider the transport of the acid into the cell. Consider the things I just talked about here. Consider also how the degradation pathway, the phenylacetic acid degradation pathway, would hook up with the cell's metabolism that's already there. So central metabolism of some sort, how are we going to hook it up to, say, either a part of glycolysis or more likely a part of the Krebs cycle slash TCA cycle, whatever you want to call it. And then we want to tally the ATP that ultimately we are hoping to make. Okay, so here's the TCA cycle. I showed this earlier, and some of its anaplerotic pathways here to keep the amounts of intermediates in the cycle where they should be. So a lot of pathways feed into the TCA cycle as well. You're not only the glycolytic pathway, but other pathways like amino acid degradation pathway, beta oxidation, things like that. Heterologous pathways can too, and ultimately heterologous pathways should feed into at least central metabolism, but preferably something like the TCA cycle. So we can have a pathway, and this is just hypothetical, heterologous pathway that we've added to, say, get rid of something. It ideally should be able to feed into the TCA cycle, not necessarily at isocitrate, but maybe anywhere, in order to get some kind of bang for its buck as it's degrading these substrates. Okay, so what you want to do, and you've already done the first step, I hope, at least if you've gotten this far you have, you want to watch this small lecture, then you want to not necessarily read thoroughly through the 3 papers that are presented on my courses in the folder that I'm about to make, but you want to skim through them and you want to take away certain bits of information. Focus on the phenylacetic acid degradation pathway itself, resulting, the results of the characterization of the pathway. You don't have to read about the materials and the methods unless this is something that really, really interests you and you really want to. You don't have the time for that, at least, and certainly I don't have the time for that, which is why I'm saying you don't have the time for that. And have access to these materials on Wednesday in class. So if you have a laptop, bring it. We will split into not necessarily 5 groups, but we'll split into groups. I have yet to do the groupings, but you will look at the groupings file that's in with the online, is in with the flipped classroom materials, and answer the questions in the handout during class on Wednesday. So you want to submit at the end of the class. You want to submit these for credit. So you will work in groups on this characterization. You have about 7 questions to answer in class. Work in groups to get the answers, and then at the end you'll submit to me, and I will grade them for credit. So if you have any questions, please email me or somehow contact me. Otherwise, I will see you in class on Wednesday, and good luck.